this. So we've covered the idea of evolution. Who could tell me what is evolution?
Is it change over time? Oh, I know.
Changing what over time? Changing what specifically?
The gene.
Yes, it's true. So it's all of this is true, but let's give it a good specific definition. Allele frequency change over time. And as we discussed in the past, what does that mean? Remember, we have two alleles, all of us, for each gene. That allele, one allele, remember we have two copies of each chromosome, right? I'm just going to go ahead and draw the chromosome like this, a single. We know that when we see them like this, they're usually doubled and connected together, but we're going to go ahead and just draw the two chromosomes this way. We know we have two chromosomes, just like the popsicle sticks that when we did the Dragon Genetics lab, right? So when we look at this and we call this, let's call it just our typical big A, little a, right? So two alleles. So how many of these big A's do you see and how many of these little A's do you see? And we looked at Weinberg, right? Does anybody remember? You'll have the equation on the test. Does anybody remember what the equation was?
P squared.
It's P squared is correct, plus 2PQ plus Q squared equals 1. And that's because P and Q represent and just to review, because it's been a good week, P is the frequency, which we usually measure in percents or decimals. So let's go ahead and put in, we're usually measuring them in percents. Remember, percents can be decimals, like 50% is equal to 0.5, right? 25% is equal to 0.25. It's the decimal times 100% would be the 50%. Does that make sense? Mm-hmm. Mathematically, that's how this all works out. So let's make sure we remember that. By the way, this is if there are things that apply to a lot of different parts of life, percentages in math, really big use, very useful in life in all forms. Taxes, tips, wages, like you need to understand percentages, guys. It's important. Fractions and percentages. Frequency of the dominant allele, right? Do you agree? The dominant allele. Now, I say the dominant allele because remember when we're talking about dominant, when dominant recessive, Hardy-Weinberg, this is a Hardy-Weinberg equation. It's usually a dominant and recessive, but you could use this equation for codominance and incomplete dominance. Where it does not work is when you have multiple alleles, like when you have A, B, and C, like A, B, O blood group, right? We have A blood group, B blood group. We'll go over that again because it could easily be on your end of course exam. Or you could have O. So 3 alleles, 1 gene, 3 alleles. This doesn't work. So this only works when you have two possible alleles, right? Then this works. And then you have the frequency of or percentage of the recessive. And when I say recessive, when we see squared, when you see this squared, is this the allele or is this what? What is that when you see squared? That's the individuals. Yeah, that's right. These are the individuals that have 2 recessive genes. So these are the homozygous recessive individuals. Does that make sense? Mm-hmm. All right. And of course, these are the individuals that have 2 dominant genes, alleles. And of course, these are what? What are the middle ones? The frequency of heterozygotes, right? Or percentages of heterozygotes. So recessive, when we talk about recessive dominant allele, we're talking about dominant allele individuals and the recessive alleled individuals, right? The homozygotes versus the heterozygotes. If we want to look at the percentages of the allele, we'd have to do P plus Q equals 1, which is all the percentages of the dominant alleles in a population versus the percentages of the recessive allele. In the population. So that's why that's where you get those two equations. One equation is talking about, this equation is talking about the different kinds of individuals in a population. And this is talking about the alleles, the percent of a type of alleles. In a gene pool. All right, so that's kind of the clarification I think some of you were asking about when it came to Hardy Weinberg. Because some of you didn't do the assignment. Oh, whoa. That's weird. No, I don't know what's going on with that, but. Wow. What is that? What could that possibly be? But anyways, so when we're talking about these equations, that's what it really means. And when we're talking about a gene pool, this idea of a gene pool, what is exactly a gene pool?
It's like with multiple different genes. Like right now, we're in the gene pool because we have different genes because was a big environment with different genes.
Excellent. That's right. So you got a group, like it could be any kind of level of group, right? It could be anywhere from the world, right? It could be a biome, some kind of biome. It could be an ecosystem. It could be a population inside that ecosystem. It could be a family, right, in the population. So this circle could be a lot of different things. It all depends on what you're looking at. When you're thinking about these things, these can all be. It all depends on the level of the problem that you're addressing. If you're addressing the problem at the world level, then you're talking about the percent or frequency of the genes on the planet. So what would be an example of that? Well, let's think about the example we used, the sickle cell. So you look at sickle cell, and we could say, what's the percent frequency of the sickle cell gene in the world? We can look at the world. Or we could look at what is the percent of the sickle cell gene in this classroom? Or we could look at anything in between. So when you're looking at these problems, you have to consider that we can break it down on many different levels. We can look at different sections and different numbers depending on what the problem or how we decide we want to look at that particular issue, an issue like sickle cell disease or a sickle cell gene. So you can look at a population. We can look at this population in this room, and we can take a look at count up how many individuals you have in here. And if each individual had 2 genes, we would put their genes in this. We just call these dots genes. And we say the white dots represent big A, and we'd say the green dots represent little a and frequency. The frequency, the percent percent of big A. For every gene in here, we have this percentage of big A, or we have that percentage of little a. So when we talk about a gene pool, we're talking about a pool, literally like a pond, right, of genes. Okay? So when we use these equations, we're trying to make sure that we're asking ourselves, what is the frequency of various genes, and what do we expect to get? What do we expect to get? We expect, if there's no evolution, that these two are always equal to 1. They're going to add up, and they're going to equal to 1, and that number is as close to 1 as possible. It's not going to be perfect, but it's going to be. If it's too far off of 1, then we're concerned. There's something going on, right? This is only true if there's no evolution. If evolution occurs, there will be a change, and may not be equal to 1. So let's take a look at an example of what we mean by change. What is evolution? Is a change in this frequency. So let's say, and by the way, how do we tell? How can we figure out how many homozygotes there are, how many heterozygotes? Well, I've told you repeatedly, and I hope that you listen, and I told you repeatedly that the coolest of dominant, when we're talking about dominant and recessive, dominant versus recessive, the cool and most important type is the recessive phenotype. Why is this the most important one for our discussions for these kinds of problems? The reason the recessive phenotype is so important is that if you see the recessive phenotype, you know the genotype. Here, with these guys, phenotype equals genotype. So we know that let's call the flowers white or blue flowers. I can look at a population, and I could see that I'm just going to put squares instead of circles just because I put a bunch of circles above. I don't want to confuse it. I got all these phenotypes here. We're talking about gene. This is a gene pool. Here we're looking at phenotypes. How many flowers do we have in the garden? We have this garden of flowers, and we have a bunch of white flowers. They're represented by white boxes. And we know, based on our studies, that the white box is equal to a white flower, which is recessive. So in other words, if you take 2 white flowers and mate them, they're always going to give you white flowers. But we also have blue flowers in the population, and that's really easy as well. And we could say the blue flowers are dominant. So what that means is if you have a big B, then you get a blue flower. Well, why is that an issue if we're looking at genes? Why is that a problem if we're looking at genes? Dominant, recessive. Why is the dominant phenotype a problem if we're trying to count how many genes? Yeah.
Because if it combines with the white flower, then it would just become blue.
That's right. That's a good point. That's part of the problem. What's another issue? If we're trying to count how many alleles we have, what's another issue? If we're trying to figure out how many big B's and little B's there are, why is having a dominant phenotype a problem? Say again? You don't know the genotype. That's right. Because this could be big B, little b, and big B, big B. We can't tell. That's exactly right. So we don't know if it's big B, or big B, little b. So how are we going to be able to tell that? Well, luckily we have Hardy-Weinberg, and luckily we have the white flower. So how does that help us? Well, if it's true that P plus Q equals 1, we know that each one of these is 2, right? But we could tell percentages right away. what percent of this population, we can't tell this, right? We can't tell P or Q because we don't know how many of the little b's there are with the blue. But we know what Q squared is, right? We know what Q squared is. What is Q squared again?
White.
The white flowers. We know the percentage of white flowers, don't we? What's the percentage? Help me out. I got 1, 2, 3, 4, 5, 6, 7, 8, 9 out of what? Count. 9 out of what?
17.
9 out of 17. Somebody do the math. What's that decimal? You have calculators. You're allowed calculators. What is it? 52%. How you got 0.52, is that right? On your calculator, you got 0.52. And then you take that, multiply it times 100%, and that gives you 52%. Do you see how you do that? That's how you go from fraction to percentage. It's important that you be able to do that. You're going to see it on your math test. You see this on SAT, guys. You took the PSAT, you saw this. You did this, I'm sure. I can't imagine they didn't give you a problem like this. I didn't see the test. I don't look at these tests, but I can't imagine they did not give you a percentage on that test. So it's 52%. So you have 52% is Q squared. But I'm going to leave it at 0.52 because I'm going to do something crazy. I don't know if you know this. You may not know this. So Q squared is equal to 0.52. How do I find Q? Does anybody know? It's a good idea to subtract because that's what you would do in algebra, right? You subtract it from one side and then you subtract it from both sides and you get one term by itself. But this is squared. How do I get rid of the square? Dividing is not a bad idea. You can divide both sides by Q, but that won't really help us because Q is a variable. So if we divide this by Q, we would get Q equals 0.52 divided by Q. That doesn't help us because then we still got the variable on the side. We want to get Q by itself equals a number. Does that make sense? Mm-hmm. So it's not that you're wrong, it doesn't get us to where we want to go. Here's the trick. Usually kids know this by now, but I'll be honest, COVID's really put us all a lot behind, right? So, but think about this. There's this thing called, have you ever seen this before? What is that? Square root. The square root is the opposite of square. When you take the square root of a square, it's the thing by itself. So what's the square of 2? Let's look at some examples. What's the square of 2? 2 squared is equal to what? 4. So the square root of 4, go ahead and do it on your calculators. The square root of 4 equals 2. So they're opposites, right? You can do 1 and you get the square of that. If you take the square root, you're going the opposite direction. So what is 4 squared? 16. What do you think the square root of 16 is?
It's just the half.
4. You know what? God, that's so easy to do. And I used to do it till I was in college. It's not half. Even though I knew it was not true, I kept doing it in my head. Square and square root are not double and half. You're not doubling it, right? It's not times 2. Doubling means times 2, right? So you have 2 times 2 is 4. The square root of 4 is the square root of 2. Square of 2 is 4. So you think that you're multiplying times 2, but 2 times 4 is 8. But 4 squared is 16. So it's different. It's different. So don't think about dividing by 2 or multiplying by 2. Make sure you keep that straight in your head. It's really something I don't think we really take the time to really express to you clearly in high school. So just make sure you understand that is a difference because it's going to affect you in every aspect of your life in high school, and certainly I think it's possible to impact you after. So if I take the square root of Q squared, then it's Q, right? Just like here, I take the square root of 4, it's opposite, right? So the square root, let me be clear by drawing it out. The square root of 2 squared equals 2. So the square root of Q squared equals Q. Is that all right with everybody? Everybody comfortable with that idea? At least the idea? Yeah. That's exactly right. You're getting rid of the exponent. And when you guys do algebra trig, and you do calculus, what you'll see is that you're doing exactly that. Is it time to go? Oh, bummer. Well, take the square root of 0.52. That gives you Q. Once you have Q, you can figure out what P is, because P plus Q equals 1. So now you're just subtracting. And now you got P and Q, and now you can figure out all the different genes in the population. Huh?
You posting it?
Yeah, I'll post this for you. Yes, absolutely. Well, somebody do that. Somebody tell me, we want to find Q by itself, so we take the square root of Q squared. That equals Q. There it is. So somebody do that. Take 0.52 and take the square root of it. If you turn your iPhone sideways, you'll see the scientific calculator. What is 0.52, the square root of 0.52? Somebody do it. I'll wait, because I'm not pulling my phone up. You guys can take the square root of 0.52. Turn the phone sideways and you'll see the scientific calculator. 0.72. All right, 0.72 is equal to Q, according to you guys. I'm assuming, I'm going to go ahead and trust you and say the numbers are right. So 0.72 is Q. That tells me what is the frequency of the little b frequency in this population if there's no evolution. If there's no evolution, remember that. That's a big if. That means if those 5 postulates are true. What are those 5 postulates? There's no mutations, there's no sexual selection, there's no natural selection, there's no advent, no gene as BB is no better than little b, right? And all the rest of those 5, it's a big population. It can't be a small one, because then you could have, you can have accidental, accidental changes in gene frequency, right? Which we call what? Do you remember what that's called? No, mutation would be a change due to, you're right, it's, no. What is it when you have a population is small and you change the gene frequency because something happens that you didn't expect? No, it can be, but it's not. We're not talking about artificial selection. We're talking about something happens, a meteor hits and wipes out a big chunk of the population. What is it? Genetic drift. So no genetic drift. Yeah, it comes from the notes that you had to study for a test that you should have got today, but you obviously didn't study. It goes back to what we had discussed the first 10, 15 minutes of class, the bad habits that we get into. We're not ready for a test. That's why we're not taking the test today. You're not going to take it tomorrow. It'll be next week, but you will not have that opportunity anymore where it's done. So, yeah, it's called genetic drift. We went over it before break. That means when the change in a gene frequency because of some bad event that only happens really easily in small populations. Large populations, you don't have that happening. So that we'll get back to that later. Bottom line is, assuming that, and also there's no gene flow, right? No little Q's coming in, no big Q's coming in, or big B's coming in, no little B's coming in, no big B or little b leaving, right? So there's no gene flow. Question?
So
Is COVID an event that could have caused genetic drift? If it wiped out a big part of the population and it was random, like if the key to genetic drift is 2 things. There's a change in the gene frequency, and it's not due to an advantage over one allele or the other. The problem with COVID is that there's an advantage to being one set of genes has an advantage over another. So that's not random, right? That means that that's called natural selection. That's where some group of people in our population are resistant to the disease, while others are not. If COVID ends up killing a third of the population have a certain set of genes, what happened to those genes? They're gone, right? And that means that there's another set of genes that had an advantage. There's going to be more of them in the next generation, right? Because those are going to be the only babies that are going to be around. and they're going to be around. Those genes will keep going. The other genes are gone or less of them. So the gene frequency has changed, so we call that evolution. So the only way we get no evolution is if that doesn't happen. Is that clear? But that's good. That COVID's a good example. All right, so of natural selection. Could someone close the door? So I'm glad we're still having a lot of giggling and talking, but wow, I guess I did waste time this first 15 minutes of class, but we're going. So so that tells me B squared. That still doesn't tell me what part do I expect, how many of these do I expect to be heterozygote, how many are homozygote. I still don't know what P is. I only got Q. How do I find? I plug it into what? No, we already had Q squared. We know what Q squared is. P plus Q equals what? Equals 1. So if I know what Q is, then P plus 0.72 equals 1. How do I get P by itself? Subtract 0.72. Somebody want to do that math? 0.35 equals 0.28, okay. All right, so we know that P is equal to 0.28. So we know that Q is equal to 0.72. If there's no evolution, then that's correct. Then the next question is, how many do we predict? This is predictions. These are predictions. How many would we predict if there's no evolution to be homozygous dominant? How many would we predict to be heterozygous? And how many do we predict to be homozygous recessive? Well, we know the homozygous recessive. We don't have to predict that because we were given that. So that number is 0.52. We know what P is, so we know 0.28 squared is P squared. We know what 2 times 0.28, let me erase that and move it over a little bit. We know what 2 times 0.28 times 0.72. Where did I get these numbers? So yeah, from all we're plugging them into the equation, right? Plus Q squared, which we already know what Q squared. We don't even have to square it because we know what it is. Q squared is 0.52. That's how we started this whole line of reasoning. So right here, this is the frequency of homozygous dominant. Somebody do 0.28 squared. I don't see. Do you guys know what body language is? Have you ever heard of body language? You know what it is? Do you want to hear? Do you want to hear what your body language is telling me? Here's the message I'm getting. Maybe you mean this. If you mean it, cool. If you don't mean it, maybe you should think about changing some of your body language. 0.078. All right, so 0.078 is the frequency of the. So that would be equal to about 7.8% is what we predict if there's no evolution for homozygous dominant. The heterozygote is what? What is this? 2 times 0.28 times 0.72. What's that number? 2 times 0.28 times 0.72, what you see on the board. It's the frequency of, that would be a frequency of heterozygote. 0.40. Let's just leave it at that. So that's going to be equal to about 40%. And then, of course, here we know, because this was given, that we have the frequency of the homozygous recessive, in this case, the blue flowers, is 52%. So what we expect is that about 40% of these are going to be heterozygous, big B, little b. About 7.8% are going to be big B, big B. And 52% are little b, little b. So how would we go backwards? What we would do is how would we test if we're right? How do we know if evolution is occurring or not? These are predicted values. These are predicted values here. These aren't actual values. This is the only actual value. We counted these. We counted how many whites. These are predicted. So now we could do genetic analysis. We can run the DNA on a gel, and we can compare them. And what we can do is we could say, I would expect this many is what I expected, the expected value. Or predicted. Another word for this is predicted. So these are the expected values. These are the observed values. And for each of these, we know that we have little b, we have little b, little b, we know is, we expected and we have 52%. There's no expected there. Big B, little b, we'd expect there to be big B, little b, we expect to be 40%. I'm sorry, that's wrong. We expect there to be 40%. And big B, we expect there to be 7.8%. In the case of our experiment, we know that there's 9 out of 17. We know it's actually 18, but we'll go say 17. How many out of 17 would we expect to be heterozygous? So what's the number expected? Number we expected is what? For the heterozygous, how many do we expect? What number? How'd you get 6.3? 17 times what? 40. 0.4. Did you say 0.4? Say that 0.4 is what was it? 6.8, you say? 6.8. We expect to see 6.8. I mean, obviously, I think you could say about 7. Can I put that? Can I put about 7? About 7. This one we know. This was given. This is something we counted. This is not what we counted. This is what we expect to see, about 7. What do we get? What do we expect? How many, when we do the genetic analysis, how many do we expect to be big B, big B? What is it? 7.8, okay. Wait, 7.8%. So it'd be 0.078 times 17. Right? Does that make sense? Why do you look? Some people are confused. Do you not understand how to do that?
0.78.
All right, so if I want to find out, if I know that 7, guys, walk through this with me. You gotta pay attention to details. If we have 7.8%, what is the total? The total is what? 17. We know it's 18, but we're going to say 17. We know that 9 out of 17, or 52%, are homozygous recessive. We counted those, yes? All right, so watch this video tonight. Take notes on it. Have a good one. Who can then tell me what would be the expected number of big B, little b, or big B, big B? What would I do to get the expected number for big B, big B? Silence is just so... No, here's the percent you expect is 7.8%. The total we have is 17. How do you find the number? How do you find the number? If I know I expect about, of the total, 7.8% would be big B, big B, how do I get the actual number of big B, big Bs in this population? I would say the expected number is equal to 0.78 times what? 17. What is that? Somebody could do the math. What's 0.78 times 17? Rennie, do you have a calculator? Oh, okay. She's doing it. 13.26. So the number I expect here is 13.26. I'm just going to say 13. Can I say about 13? So that's the expected value. All right? I expect about 13. How much would you round off? What's the significant digits? We can figure all that out, but it's really not important for this case. Yeah.
So how are you saying that there's 17 total in population? Then we got 13, 7, and then 9. That's more than 17.
You took 0.78 times 17, you got 13? And 0.52 times 17 is what? That number should be a lot less. Yeah, that seems wrong to me. What's 0.52 times 17?
8.84.
So, and what's 0.78 times 17? It should be less than 9.
It's 13.26.
No, there's something wrong. I have to do the math myself. Hold on a second. That doesn't, I'm not saying that you're doing anything wrong. It just doesn't make sense. 52% of 17 versus 7.8%. Did you multiply 7 times? No, I can't. So you did 0.078, or did you do 0.78?
You told me to put 0.78.
Oh, I meant 0.078. Sorry. 0.07. Do you understand the difference? One's 78%, the other's 7.8%. Right? That's the problem. So what's 7.8%? Go ahead and do that number again. I did. I even wrote it here like an idiot. It's not 0.7.
1.326.
What is it?
1.326.
So 1. It's the numbers are equal to about 1. We expect to see about 1 big B, about 7 big B, little b's, and about 9 little b's. Everybody see that? Yes?
Yes.
Okay. All right. So how many do we observe? What if we saw 9? What if the percentage? What if the number here's the number we observed was actually? What if we saw 10, and we saw 8, and we saw 2? It's equal. I don't even know if that equals more than what we. Sorry, that doesn't work. What if we saw? I have to do this in my head. I apologize. I should have done it earlier. But let's see if we saw 8. No, let's say 7, and we saw 8 and 2. Now, is this evolving? This is what we actually see. And the question is this evolution, right? If we see this, and this is what we expected to see, is the difference between these two evolution. Correct? Understood? Yes, no? Yes? And unfortunately, I can't do these numbers. These numbers would work, but I can't use them because we said we saw this, so we have to keep this the same. So this is 9. But what if this was, I'm trying to do this in my head. What if this was 3 and this was 4? Is this evolution? Or now the numbers work out, right? 9 plus 4 plus 3 is what?
16.
Did I do it wrong again? Oh, it should have been 4 and 4. Yeah, I left out 1. All right, now we have 17, right? So out of 17 total, what we see is 9 of these, 4 of these, and 4 of these. Is this evolution? Is this close enough to this? Is 7 close enough to 4 that the difference is random error? Or is this different enough that we have to say there's evolution? We have to do chi-square. So you have to do chi-square to really answer the question. So how do you do chi-square? How do you do chi-square? Who knows what the formula is?
P squared.
That's Hardy Weinberg. observed minus expected squared divided by. Is this right?
Summation.
That's right, because it is a summation. Summation of what? Well, this would be for the little b. Then you add it, right, to what? To the same thing for what? So I have a lot of silence in the room, and I have to ask the question, are people not understanding? And there's silence again. It's interesting. What's the answer? Do you understand this?
I want to.
Okay, is there a problem? What's the problem? Why are you not asking questions? What part of this don't you get?
I don't know what questions to ask.
Do you understand how we got the numbers here? How did we get these numbers?
Using that equation.
No. What are these numbers? Who can tell me what these numbers are?
The number of flowers in a population.
Yeah, no. These are observed. So we took the flowers and we did a genetic test. We ran their genes on some kind of electrophoresis machine, and we separated them. We identified who was heterozygous, who was homozygous, and we knew who was the homozygous recessive. How do we know how many were homozygous recessive? One more time, repeated from earlier today. How do we know the Q squared? How do we know which one's homozygous? How many were homozygous recessive? We can count them because homozygous recessive, you see what you get. The other two, we had to actually analyze their genes. And what I'm saying to you is that what we observed, what we expected, according to Weinberg, yes? Yes? In the back? Yes? I want to hear it. Yes? Okay. So, I mean, if I have to be like a preacher up here and demand, like a coach, and demand to hear yes, then that's what I'll do. All right? So, according to Weinberg, these are numbers that are true, should be happening if there's no evolution going on. All right. So if these frequencies hold true, if there's no evolution going on, and I plugged and chugged, I did the math, anybody have a problem with that?
So if it's evolution, we gotta use chi-squared.
No.
Oh.
We have to use chi-square. Okay, so listen to what I'm saying, please.
I'm listening.
Look at me in the eye.
I'm listening.
Okay. We counted.
Right.
How many recessive phenotypes there were? Yes? That gave us what? Q squared, right?
Right.
So allowed us to predict how many Qs would be found in the gene pool, correct? Mm-hmm. Which allowed us then, using this math, to predict, based on that, we were able to predict how many we expected to be homozygous dominant, how many we expected to be heterozygous, how many we expected to be homozygous. Well, we knew this one. We knew this was because we could observe it. Are we clear? I'll keep asking until I get an answer. Are we clear?
Yes.
Because I'm going to expect that you answer. I'm going to expect it. Because obviously people are not participating mentally. All right? So here we go. This is what was predicted. So how do we find out how many? These are the percentages that were predicted. This is the percentage that was observed. 9 divided by 16 or 17 gave us 52%. These percentages were predicted based on the equation, correct?
Yes.
Okay. How would we find the numbers expected or predicted? We took those percentages and multiplied them times the total. Yes? Yes. Yes in the back? Yes.
I have a question.
Yeah.
So how are the percentages not adding up to 100, but they adding up to 17?
Wait, what? These are about. I'm rounding off. So it's a good question. These are all quickly calculated numbers. Okay.
Why
This
don't
is.
we-
Say what? Go ahead.
Okay, never mind. I see why now.
Yeah, like if you took 7.8 and you made this 8 instead of 7.8, then 8 plus 2 is 10. That would be 60. 60 and 40 is 100.
Okay.
But we're not. When we came up with these numbers, we rounded off. That's why I have this little symbol about 7. Right? It doesn't make sense that where I'm going to have 7.2 individuals, right? That wouldn't make sense. So I just rounded them off.
Okay.
All right. It's a good question, though. Excellent. That's the kind of question I wish people would answer and ask in the back and in the sides and in the front, rather than just sit there quietly and force me to actually demand your participation. Here's the observed values. These are what we observed. In other words, I counted these. Emily, how did I get 4? How did I get 4 big B, little b's? Where did I get this number? What do you think, Emily? I told you the answer. I told you all these answers already, but obviously I had to go back and slow down a little bit. So where did I get that number, Emily? Do you not know? Just say, I don't know. All right, thank you. So the number I got, where did I get 4? I took all the rest, all the blues, right? I couldn't tell what the blue flowers were. Why, Emily, why could I not tell what the blue flowers were, whether they were heterozygous or homozygous dominant? Because they're all blue. That's simple. So what did I do? I had to break their cells down and run their cells on a gel and separate the DNA and identify how many of them were homozygous dominant and how many were heterozygous. Does that make sense? So these are observed values, but I did it in a lab. Are we good?
But like on a test, they're not gonna say you did it in the lab.
No, they're going to tell you that. They're going to tell you I ran the gels. We ran the science. What they'll say is on the test they'll give you the numbers. Yeah, they'll give you the numbers. They're not going to expect you to figure it out. You
So
can't.
It doesn't really matter how they got these numbers. It's just you're gonna be given.
It might matter because they usually ask you. They'll usually give you an experiment, and on FRQ they'll ask you a little I, 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 etc. Right? So it might matter because they might ask you how would the scientists have found these numbers? Right? They could ask that, and you'd have to know. Like these are genes. How do you find what genes something has? You have to do genetic analysis. You have to run it on a gel and look at the gel and identify which has the big what version of the gene. Right? Does that make sense? So it does matter, in a sense. But for this problem, it doesn't. It's just important that you understand that they're observed. So 944 is what's observed. Does that make sense? That's why I had to change the numbers a couple times. I did it off the top of my head and it didn't come up to 17. I want to make sure that these numbers came up to 17 because there are 17 individuals out there. So the question we have is back to the meaning of Hardy-Weinberg. Why is it important to find out if these numbers match these numbers? Ready? Why is it important answer that question? Do these numbers match, and are they close enough to these numbers? Why? Right, so tell me, what does that mean? That, by the way, that's an easy FRQ question. Easily a little I, little I. What does it mean? What are the possible options here? Tell me. If they do match, if they are close enough to each other, then what? If they're not close enough to each other, then what?
If they're close enough, it means that there isn't evolution going on. And if they aren't, then there is evolution.
Exactly. So if the numbers are close enough to the predicted values of Hardy-Weinberg, then the difference between them is not significant, and therefore there is no evolution going on. That is correct. There is a difference. Everybody agrees there's a difference. The question is that difference significant or not? Is it big enough? We can't answer that off the top of our heads. You can't. It's not possible. So let's go on and look closer then. Let's look at the equation. So what is this? So that's why we use chi-square. Chi-square answers what question? Who should I pick? Who can tell me what, I'll ask in general. Who can tell me what does chi-square answer? Why am I doing chi-square? Yeah.
See if the outcome is close up to degree of freedom to tell there is evolution or not.
Exactly. Perfect. To see if the difference between the two is significant. If it is, then if it is a significant difference, then I have to reject the null hypothesis, which means I have to reject that there's no evolution going on and accept that something else is going on, that there is evolution. Does that make sense? Okay. All right, so the observed number for each of these, I'm going to go ahead and put the observed numbers in red and the expected values in white. So the observed value for little b was 9. The expected was 9. I square it and I divide it by 9. For the next one is a heterozygous, so that would be 2p squared, right? Or I should just put big B, little b, I guess. Big B, little b. And the last one then, of course, is big B, big B. So big B, little b, I observed 4. I expected 7. I square it and divide by 7. And then lastly, for big B, I observed 4. I expected 1. I square it. I divide it by the expected. Oops, which is 1. Well, the values are, this is all together summed up. That's chi-square. So obviously 9 minus 9 is 0. 0 squared is 0. 0 divided by anything is 0. So this is 0. To be clear, if the bottom number is 0, then it's undefined. It's, if you remember calculus, it's towards infinity, right? So 4 minus 7 is what? 3. Negative 3, but 3 squared is negative 3 squared is 9. 9 divided by 7. And 4 minus 1 is 3, and that would be, again, 9. And that would be 9 divided by 1. So add these together. 9 divided by 7 is what? What is it? 1.28. Plus 9 divided by 1 is 9, which equals 10.28. Well, that's great. That's chi-square. But I have to compare that to something. How do I know? That doesn't tell me if it's significant or not. So I have to do something with this number. I have to do something with 10.28. I need a table. Now, in a problem, you would get a table. In a problem,